Hello and welcome to this week's What Were They Thinking? Among the range of news items we have for you this week, ubiquitin's ubiquitous role and its use stopping the immune system from attacking otherwise healthy cells, otherwise called autoimmune diseases. We have a somewhat significant development in treating brain tumours in days, rather than the weeks, months, and arguably even years it would otherwise take. The beneficial results of COVID-19 with regards to heart failure and blood clots. And yes, this will trigger every single anti-vaxxer that has spouted bullshit about the dangers of vaccines. Possibility of creating insulin using cow's milk rather than the current method that relies on genetically modified bacteria. This drastically increases the volume of insulin that can be produced. A novel way of using yeast that would stop some of the more aggressive and invasive fungal species that are causing disease. One of the oldest polio survivors has died after having spent 70 years inside an iron lung. Your time on a computer might be contributing to your erectile dysfunction. Orcas are being more and more creative, and unfortunately, they're using this creativity to improve their hunting techniques. And we have a effect of misspelling cell lines in academia that leads to all kinds of issues, but particularly the creation of otherwise non-existent new cell lines. The timestamps for these and other news items can be found in the description box below. Let's start with the role of ubiquitin in handling the immune system and, at least theoretically based on this research, contributing to the management of autoimmune diseases. As said earlier, ubiquitin is a ubiquitous and a useful molecule within the body. It has many roles to play, but its major job is in the handling of proteins, either the breakdown or otherwise controlling the activity of them. For example, ubiquitin has a major role to play in the human brain development as a fetus and then later as a child, notably in the pruning of neurons. This means that ubiquitin contributes, at least somewhat, to things like autism, Angelman syndrome, and so on. But it's not just neurons. As noted with this research, it can also have an effect on things like the immune system. The way this research proposes to be able to use ubiquitin is that it's going to be targeting an enzyme called cyclic GMP-AMP synthase, otherwise simply given the acronym CGAS, although it's a terrible acronym since acronyms should not contain other acronyms. Beyond that pet peeve, what's important here is that the cyclic GMP-AMP synthase has one job. Its role is to bind to any DNA floating out of the cell cytoplasm, and basically any DNA not contained within the nucleus. This would typically be due to pathogens, for example viruses. However, on rare occasion, the cyclic GMP-AMP synthase decides that this is not what it's going to do. It begins to act out of its role, and when this happens, it begins to cause issues. And it begins to mark areas and places where you should begin to break down protein that there should be no protein broken down. This stimulates the immune response by controlling the activity of the synthase, and by allowing ubiquitin to play its job, both to shepherd it away from where it shouldn't be, and by preventing the cell's immunological processes being stimulated, thereby activating other more systemic immunological responses, you avoid the issue of autoimmune diseases. This is something, for example, like diabetes, where the immune system, as a very young child, begins to target the beta cells responsible for the production of insulin. If, and this is a massive if, you could stop these responses from occurring when a child is, well, a very young child, you could theoretically stop type 1 diabetes in its tracks. Of course, this is all theory and very much based on cell studies. What is not based on cell studies is understanding what sperm does once it gets into an ovum or egg, and how it prevents other sperm from getting in. Effectively, curse your sudden but inevitable betrayal. There are almost no known cases, and we say almost no known cases for a reason, of semi-identical twins, because there are only 2 instances where a sperm and then another sperm got into an ovum. 2 cases in basically all of humanity's knowledge. That's not really a pattern, that's some weird exception. But how does the sperm stop that from happening? There are a couple of models out there, one involving calcium and a cascade preventing the ovum from being permeable, but there's another method that's been proposed, and it's a membrane protein called ZP2. ZP2 is supposedly, from this research, cleaved or broken into multiple parts after the sperm enters the ovum, and because it's broken, it makes the ovum coating or shell harder and therefore impermeable to the other sperm. This research may sound rather pointless, but there is a good reason why they're looking into it, and it all comes down to fertility, and to a certain extent, success of fertility treatments. It may contribute, and to be clear, may, given that at this stage we're not entirely clear about what's happening, it may contribute to the ability to make ovum more likely to accept a sperm and more successfully continue on developing after that. On the other hand, it may contribute to, the exact opposite of fertility, contraceptives, by, if not chemically, at least hormonally triggering the ovum into thinking there is already a sperm inside of it, thereby preventing the issue of any sperm being able to get into the egg during intercourse. Effectively, the same idea that's used for the current oral contraceptive regarding estrogen and progesterone, which primarily act on the ovary rather than the ovum. Moving from the start of life to what for many is the, end of life. It's a brain tumor, but in this case, theoretically, delaying the end of life. Although at 72 years old, whether or not you want to pursue life may or may not be desirable. The 72-year-old man had a rather nasty kind of brain cancer, and we say nasty because it's not only aggressive, but the location makes it really hard to treat. When you get a tumor in the brain, it's very hard to get medication to it, owing to the blood-brain barrier and the fact that it's rarely very easily penetrated by most drugs. It's just not permeable, which is what you want most of the time. After all, the more things you can get through the blood-brain barrier, the more likely you are to have issues with regards to pathogens or unwanted content in your brain. For example, music that just won't go away. In this case, chemotherapy won't do much for the music, but it will do something for a glioblastoma. In this instance, the cancer will pretty much kill anybody that has it within 5 years. So we're talking about a pretty much death sentence if you're diagnosed with it. However, for the 72-year-old man who received this treatment, it was almost entirely gone. However, the other 2 individuals who received the same treatment weren't so lucky. So while a 1 in 3 chance of effectively having the cancer entirely go away is great, it's still not good enough, mostly owing to the inconsistency of the outcome, and understanding why that happened would be a major achievement. The major thing around this treatment is, first of all, it's not a chemotherapy or similar drug-based treatment in the terms of what you might think of as drugs. Rather, this is relying entirely on the immune system and specifically on the T cells. T cells are meant to be programmed to attack pathogens, but they take time to be trained to identify them. But if they never get trained to identify them, you never get the T cells made and therefore they don't destroy them. This is where the T cell treatment is particularly useful. They can get into the brain. Admittedly, it takes longer, but they can do it. This means that you can give a patient T-cell treatment and you can begin targeting it, but you don't even necessarily need to use the T-cells. Instead, what you can do is get B cells to make antibodies to the specific tumor, and then you can attack the tumor that way. This will then trigger the T cells to activate. It is worth us pointing out that both of the other individuals who were given the same treatment did initially show very promising results with a significant reduction in tumor size. The problem is, for both of those individuals, it would appear that the immune system didn't take to the particular treatment, and therefore, although initially reducing the size of the tumor, it then started growing again later on. This means that, at least initially, this treatment has promise, but it's nowhere near ready to move on from its stage 1 clinical trial. Comparing this to something like the RSV vaccine is not very favourable, but it is important to have data, and we do have data on the RSV vaccines that have been given out in America for about a year now. RSV is a rhinosinusitis virus, and it's a particularly problematic virus for infants and toddlers who are the most likely to be affected by this. Older children and the elderly can be to some extent, but you don't really see it with them as much. We now have numbers, at least in America, about how well it's worked, and the numbers are incredibly promising. In short, the number of children vaccinated and the percentage of those children going to hospital because of RSV has dropped between 80 and 90%. Those numbers are great, except there's one major problem with them. The percentage of children who are actually getting the RSV vaccine is very small, and that's because parents are declining it, either because they're stupid or because they've been convinced of the lack of necessity. The third possibility, although arguably less likely, is that given that this is American medical care, you would have to sell several organs to be able to pay for it, and they've already given up their liver, one of their lungs, and probably their kidney just to be able to pay for the medical care of the baby so far, so they don't have anything else to sacrifice. Another vaccine news, but this time news that's going to piss off the anti-vaxxers who keep spreading bullshit around the effect of the COVID-19 vaccine and how it's causing blood clots and other issues. Recent research from Europe has found that actually there is a relatively little risk, and certainly a much less risk, if you receive the COVID-19 vaccine for blood clots and heart failure. Those things that keep being brought up by the anti-vaxxers who say, look at this bizarre blood clot that they found, and it turns out it's actually a bullshit, either photoshopped or misappropriated picture. The data is based on a large cohort, and the University of Oxford was looking at it. The records for this large cohort came to 10.2 million vaccinated individuals and 10.4 million unvaccinated people. Although, to be clear, the unvaccinated was naive data, so this is before they were given the vaccine. Further to this, it's not just based on United Kingdom data, but also Spain and Estonia. So we have a rather large portion of European continent covered by this research, so you can't claim that there's going to be particular geographical issues. Of note from this study, we have ischemic stroke, hemorrhagic stroke, transient ischemic attack or temporary stoppages in blood flow, ventricular arrhythmias, cardiac arrest, myocarditis or pericarditis, a myocardial infarct, heart failure, pulmonary embolism, deep vein thrombosis, venous thromboembolism, and a couple of others that all got examined. Most of the time, when you hear of the COVID-19 vaccines, you'll be hearing of things like a myocarditis or pericarditis, or in some cases, things like embolisms and a myocardial infarct. These are the major anti-vaxxer talking points, and in short, to spoil the results, they're all more common in those that are unvaccinated. Talking about some of the most extreme reductions, we have things like a 72% risk reduction for venous thromboses. That is a clot in your veins. For heart failure, we have about a 22% reduction. The other, let's say, powerful aspect to this study is that they were able to identify each of the different kinds of vaccines given to people, and therefore break down the different effectiveness on vaccines with regards to reducing the risk of these events. And although there was some difference in about the first month of vaccination, the study found that regardless of which vaccine you received, there wasn't much difference after that first month. Therefore, not only do the vaccines work, but they provide other benefits. By contrast to this, we have the effects of COVID-19 on your brain and the fact that there are, at least according to one study, notable drops in the IQ scores. Yeah, getting COVID-19 will literally make you dumber according to this research, although, again, take it with a lot of skepticism just based on what it is. IQ is not necessarily a great measure of anything, given that those who score well on IQ tests tend to do well at IQ tests, but aren't necessarily effective at other activities by comparison. The Scientific American is effectively providing an article that summarizes not just some of the major studies, but the effects of COVID-19 from these major studies. For example, risks of cognitive deficits or decline in cognitive function, that there are issues around brain volume, that there is brain aging, and so on. We won't go through all of the various effects, but it is worth noting that there is a fairly long list of not just articles that have found this, but the effects that COVID-19 has with regards to cognitive function. Shifting from diseases to treatments, but this time treatments that straddle the line between genetically modified and naturally occurring. In this instance, it's the ability to engineer cows so that rather than having to rely on genetically modified bacterium, the cow can make milk that contains insulin. It might seem like an odd thing to do, but there is a very good reason why they want to make it this way. Your traditional approach, or at least traditional in terms of its being the primarily preferred method for the last 50 or so years, is to take bacteria, have 3 vats that will each produce 1/3 of the insulin molecule each. Then they effectively drain the vat, separate out the bacteria from the insulin, combine the insulin parts in a separate container, and this therefore creates the insulin that is used. It's rather labor intensive, requires a certain amount of expertise, and as mentioned, it has 3 separate processes involved. Not exactly ideal. It also doesn't produce anywhere near as much insulin as this approach would by comparison. To understand this, the single liter of this milk could produce enough insulin for a single individual for 8 years, but this assumes that you could scale up otherwise reported research and it would be applicable to the cows, as they haven't actually published how much insulin was produced, which is annoying, but nonetheless. But beyond just the possibility of the insulin, there's the fact that, well, you couldn't use the milk itself as a source of treatment, as the digestive acids within the stomach would deactivate or denature any kind of attempt to deliver insulin that way. But it does mean that you could have a relatively small herd of cattle that, when milked, could produce more than enough insulin for a country. And we say a relatively small number, as you would only need 100 or so head. In other similar genetically modified animal news, we have the first genetically modified pig kidney being transplanted. There has been research over the last few years, in fact it's probably closer to say 20 years, to be able to take pigs and genetically modify them to remove the various immunological triggers that are contained on the surface of the organs. This therefore allows the organs to be transplanted without having to worry about the immune system attacking them and destroying them. an important thing in cases of organ shortage, especially from donors, which is pretty much everywhere in the world. The 62-year-old man from Massachusetts had end-stage kidney disease, and he received the organ only a few days ago, last Saturday to be precise, and it took about 4 hours to give him the kidney. Based on the state he was in at the time of the article that we got this from, he was recovering well and expected to be discharged in very short order. The importance of this development cannot be understated, especially in those countries where there isn't a opt-out system for organ donations. That's because the number of organs that are donated is tiny compared to the number of people who need them. It's something like 1 donor for every 87 people who need one in America alone. It's ridiculous, and arguably, it's each person's choice, but you're not going to be using that kidney once you've been put in a box 6 feet under. Nonetheless, solutions had to be found, and since opt-out systems weren't going to be put forward in America, they've chosen an alternative. This was to find another way to get more kidneys. More kidneys so that you don't have to have everybody sitting on dialysis machines before too long. The new kidneys, as mentioned, come from pigs, and by genetically modifying the pig, it's not just the kidneys that could be used. In theory, you have the liver, theoretically the heart, the lungs, the valves of various parts of the venous and arterial system, and more again. In further treatment-related news, we have the role that a new yeast strain might have in being able to deal with rather invasive fungal infections. It is ironically, a collaboration between Israel and Germany. The new form of yeast, named Kazakhstania weissmannii, is able to help the body in dealing with Candida albicans. Candida albicans is one of the major pathogenic forms of yeast and responsible for things like thrush. The way it works is that the 2 forms of yeast effectively have an adversarial relationship, and so one will keep the other in check and thereby prevent either from becoming pathogenic. It is worth us pointing out that much of this claim is based on the idea of being able to inoculate mice with Candida albicans, but that some of the lab mice within the facility they were using simply could not be colonized with this particular form of yeast, and that they found that even though they were eradicating, theoretically, all of the bacterium within the GI tract, it was down to the presence of a, at the time, unidentified form of yeast. And next we have news from Florida that is disconcerting. And you should be mindful that if you happen to be hearing a scratching from inside your head, perhaps take a trip to the hospital and ask them to take a look inside. The Florida man, who was 52 years old, was sent for a CT scan to have a look at his brain. Sadly, not for doing Florida man things, but for being ill. What they found was a tapeworm from pigs inside his brain. Well, we should be slightly more clear. The tapeworm itself wasn't in his brain, rather cysts from the eggs of the tapeworm were inside his brain. The challenge with this Florida man is not so much the diagnosis itself. Treating it is possible. You use antihelminthics. Rather, the issue is that the human body does not respond well to treatments for the brain. This means you need to do 2 things. You actually need to lower the dose of the antihelminthics, and you need to provide anti-inflammatory drugs at the same time. If you don't do both of these things, you effectively trigger the brain and, well, you set it on fire, and you pretty much get this. The problem with that is, well, it's not good for the individual or the brain, leading to complications. Doing both will allow for the reduction in inflammation, and although not pleasant, it doesn't result in anything as dangerous. In terms of treatment, we have one of the longest running survivors of polio, who's now unfortunately died after having spent effectively 70 years in an iron lung. Now, there are various individuals out there who will claim polio is not dangerous, you'll be fine. And then there are others who claim that, oh, iron lungs aren't needed anymore because respirators and ventilators. But no, unfortunately for you, that's not true. Unfortunately for us, it's not true. The individual from Dallas, Texas, got polio at the age of 6, and, well, one of the symptoms of polio is to be paralyzed. A lot of the time it's a leg paralysis, or at least that's the most common form you would see. But in this case, this poor bastard got paralyzed from the neck down. The individual in question, however, was somebody who should be looked at as a model of somebody who achieved things despite challenges, given that not only did they get a law degree, but they worked in the legal field too, and despite being paralyzed from the neck down. Although polio is largely a disease of the past, it is seeing something of a resurgence, unfortunately. We have modern diseases to worry about too, and it's a relationship between time spent staring at a computer screen and erectile dysfunction. And given where you're spending your time using that computer and how, erectile dysfunction might not be exactly what we'd expect. Instead, staring at TikTok videos and YouTube videos could be contributing to this. It is worth us pointing out that the research comes from China, and China does have some, severe limitations on what people can and cannot access, but also really weird laws, particularly around time children can spend on a computer. It's not to say the research is necessarily wrong, just that there's a lot more complications to research like this conducted in China. What they were trying to do was something called Mendelian randomization. The idea was to see if there was some sort of correlation between certain tendencies and the certain genes that were involved, and see if there was a pair relationship between them. One upside to this study is that rather than relying on Chinese data, which as mentioned has all kinds of issues, they instead looked at 220,000 records of European individuals, which does help somewhat in mitigating the problems, but this does also mean it's a purely observational study and therefore can establish a theoretical correlation, but it can't show anything definitive. What they found was that every 72 minutes you spent watching TikTok videos meant you had a 3-fold increase in the probability of erectile dysfunction. Well, it didn't have to be TikTok videos, it could have been any kind of leisure type activity involving a computer. Well, we'd say any kind barring a couple that should theoretically have the opposite effect of erectile dysfunction. In news that's likely to be obvious to anybody, the World Happiness Rankings have come out and the US has dropped drastically in its position. The USA has been in 8th position last year, and now it's not even in the top 20. Which, given the way the world is, and particularly America, people not being happy is really no surprise. It's kind of odd that Finland is the happiest place in the world, but nonetheless, it's strange that America, or for that matter any other countries, are happy. Where these numbers get kind of interesting is they've also published an alternative way of looking at the data, and that's based on age brackets. For those under 30 in the US, they're now in 62nd place. This is compared to countries like Saudi Arabia and Guatemala that are higher ranked. Yeah, people in Saudi Arabia, if they're under 30, are happier than those in America. Which again, probably isn't that much of a surprise. In America, where you're swimming in debt, you can barely afford to eat, and your rent takes away what little money you do get, yeah, not being happy is no surprise. Shifting to animal news, although animal news that would be in some ways just as disconcerting, depending on how you want to look at it, orcas, or killer whales, have found new ways to be able to hunt effectively. The orca, other than being a giant carrier for the mammoth, is a whale-like creature, although technically speaking, it's in that weird place where it's not a true whale, but it's also not a shark. It is still a mammal. Because it's a mammal, it has all kinds of issues around its life, and particularly being an aquatic creature. Therefore, it needs to be able to get oxygen, get food, and reproduce. Food is the challenge that they face, and the population up in the Northeast Pacific have found that there are two sources of food that are surprisingly convenient and tasty: seals and baby whales. Getting either of these sources of food can be a challenge for the orca, and particularly if it's going to take the same approach you would typically imagine for a predator. Think of how a lion, tiger, a wolf or similar catches its prey by getting its jaws on it and holding it. Well, the orca has one option that these animals don't have. The orca is big, it can be very fast, and it's in water, meaning it engages ramming mode and quite literally rams into the prey or slaps it with the fins or tail, effectively knocking it out before then consuming its prey. Of course, these are not the only methods available. They've used a range of other approaches to deal with the dolphins that are out there, porpoises, and other sea creatures. Some of it seemingly rather brutal, but also at the same time rather effective. In mathematics news, we have a man who won the Abel Mathematics Prize. And what is key here is that the guy's turned to mathematics and won his prize out of necessity rather than out of a particular desire to do so. The man won the Abel Prize for his work having to do with random behavior and mathematical modeling of it. Now, it's worth us pointing out that the Abel Prize is effectively a counterpart to the Nobel Prize, specifically for mathematics, because, strangely enough, the Nobel Prize doesn't offer a mathematics option. It's physics, health, peace, literature, but not math, which is, as said, odd. So, the Norwegian government has made up for that issue, let's say, with the prize. The guy who got it is arguably not necessarily inspirational, but someone you could look to as a sort of role model. Why he's turned to mathematics out of necessity is that he had concerns he would go blind. He had multiple issues to deal with retinal detachment, and so, fearing he would go blind, opted not to enter into other fields, but followed his father and became a mathematician, and eventually won a effectively Nobel Prize equivalent. In other research-focused news, we have, the reality of what happens when you misspell something, and specifically, misspelling cell lines in published academic work. You'll find that most immortalized cell lines that are used for biological research tend to be given particular names. For example, HEK, and then a series of numbers, generally 3 numbers. These would be a immortalized kidney cell line. But the problem happens when you either get that name wrong, or you just make up a name. This is where things get to be difficult. In particularly cancer-based research, there is a growing prevalence of this exact phenomena. That is, cell lines are being given either the wrong name, due to issues around misspelling them, possibly autocorrect as well, or for others that they're simply not following conventions. It's not just this though, there's another problem, and along with cell lines not being named correctly, we have that, to be polite about this, there were misspellings around the gene sequences that were being discussed. This is a problem, not just because it means that if you were to try and search for these gene sequences or just sequences in general on the various platforms that are available. Consider BLAST from the NCBI service, the American government's National Institute of Health. You would not be able to find these because they don't exist on there, because they've been misspelled. This is the problem. You wouldn't be able to recreate the research that has been done because the research has been improperly described in the literature. Effectively, the problem with this phenomena is threefold. One, it means replicating the research is nigh on impossible. Two, trying to conduct further research is nigh on impossible, simply because you can't replicate what you can't get or find. Third and final, you don't actually know what the foundational research leading to that publication was, meaning that there is no underpinning developments. And since most research is meant to be built on top of prior research, effectively thinking of it as bricks in a wall, slowly building itself up until you have your entire structure, that being the entire field of research, you can't even start at the foundation because you don't know where that is. The final news we have for you this week is a star that's going to go supernova this week. Although, strictly speaking, it's already gone supernova about 3,000 years ago, but we might be able to see it now. NASA has told us that this is likely to be a once-in-a-lifetime event, and you can see it with the naked eye for up to a week, given how long the process takes. To be able to follow this, it's either worth following the NASA Universe account on X/Twitter, or if you want to be able to locate the general location of where it's going to happen, you want to find the Hercules constellation, and you'll find that there is a group of 4 stars in the center of it. Those 4 stars are roughly where you want to look to be able to see this occurring. While watching in the hours just after sunset, you should be able to see it occurring as a flashing light. It's worth pointing out that you will also be able to see a solar eclipse, at least in Canada and North America, throughout April as well. So depending on when you go look for this, you might be able to experience a variety of, let's say, astronomical events of particular interest. That's all the news we have for you this week. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please do post any comments, questions, or suggestions you have below.